Hi everyone, this is our third mini lecture in Chapter 13. I will be talking about antigens and antigen presentation. When we talk about presentation of antigens, we need to discuss the entrance of these antigens, or immunogens, which is synonymous. They're substances that can elicit an immune response. So they have certain requirements that they're going to have to meet. Obviously, they're going to have to be foreign or have foreignness. Their shape and size and accessibility is also going to play a role in what kind of a response they elicit. They cannot be a normal constituent of the body, so they must be something other than self. And examples of this would include a whole microbe, perhaps a bacteria or virus, or their parts. Also cells or substances that arise from other humans, animals, plants, and then also various molecules, which could be chemicals that are introduced into the body. Complex proteins and protein-containing molecules are much more immunogenic, so elicit a bigger immune response than repetitious polymers composed of a single type of a unit. And I'll go into that a little bit more. So most antigens will fall into the following categories. We have proteins and polypeptides, and these would include things like enzymes, cell surface structures, and exotoxins. Lipoproteins would be another, which often are components of cell membranes. Glycoproteins, which can be blood cell markers. Nucleoproteins, these would be DNA that are complexed with proteins. Pure DNA would not be recognized as foreign, as DNA is universally the same from organism to organism. Polysaccharides and certain bacterial capsules would be made up of this. And then lipopolysaccharides are other ones that would fall into this category. So when we look at a comparison of good immunogens versus poor immunogens, you can see that we have microbial cells and viruses, perhaps plant molecules, and then foreign human or animal cells, for example, blood from another human being. You'll see that proteins have many of what are called epitopes. These are the binding or the part that will be noted to bind to a receptor and recognized as an immunogen. When we look down here at poor immunogens, these are things like a trisaccharide, so 3 of the same sugar in a row, or maybe a polypeptide that doesn't have any real unique features that would make it stand out. So when we look at those more complex of the immunogens and the ones that elicit the good immune response, we're looking at the epitope of it, which is the portion of that antigen which a lymphocyte will respond to. And it's the primary signal to the immune system that the molecule is foreign. So it's what that lymphocyte will see as foreign. Now, we have something called haptens, and these are small foreign molecules that are too small to elicit an immune response. And so what ends up happening, in order to get an immune response, it needs to be linked to a larger carrier molecule. And that combination of this hapten with a larger carrier molecule will cause an immune reaction. And at that point, the hapten will serve as the epitope. And this is just an example of that. We have what looks like a disaccharide, not very good immunogen. It gets attached to something, a carrier molecule, and at that point it would be a good immunogen, and the hapten is what would be the epitope of this molecule. So we also have something called alloantigens, and these are cell surface markers or molecules that occur in some members of the same species, but not in others. And this is a good example would be an individual's blood group. So when we talk about A, B, and O blood types, we're actually looking at the alloantigens, and it would be the basis for that individual's blood group and major histocompatibility profile. And it is what is responsible for those incompatibilities that can occur in blood transfusion or organ grafting, and why we look to make sure that it's a match ahead of time. We also have something that is called super antigens, and as you can imagine, these are antigens that cause a much greater immune response than a normal antigen. And these could be bacterial toxins, and in that case they are a potent stimuli for T cells. When they activate these T cells, it's at a rate of 100 times greater than just an ordinary signal. And this can result in an overwhelming release of cytokines and cell death. Now, it can be detrimental at times because sometimes it's almost like an overreaction. And we can get toxic shock syndrome, and also certain autoimmune diseases are associated with superantigens. And you can imagine why this might be true, since it elicits such a strong reaction. And then allergens, which we're not going to discuss in this chapter, these are antigens that will start an allergic reaction. And these are discussed in chapter 14, which we won't actually be covering, but is a very interesting study. So what actually happens? How do we have these antigens presented? In most of the immune reactions, the antigen is going to be acted upon and then formally presented to lymphocytes by what are referred to as antigen-presenting cells, or APCs. And some examples of these would be macrophages, B cells, and dendritic cells. And what ends up happening is they can process this antigen, and then they're going to present it. So the antigen will be bound to the MHC receptor that we talked about in one of the last lectures, and they are moved to the surface of either the macrophage, B cell, or dendritic cell, so that a T lymphocyte can come and recognize it and bind to it. So let's see what that looks like. This is table 13.3, and we see up at the top there's a microbe that is grabbed or engulfed by an antigen presenting cell. This could be a macrophage, a dendritic cell. It's going to be broken down and processed in some way, and then it's going to have part of it displayed or presented by our MHC2 receptor. Okay? And this is a point where a T helper cell can come along. So that MHC2 antigen will then bind to our T helper cell. It comes in and it recognizes. And what ends up happening is that the T cell receptor is going to bind the antigen. And our CD4, remember we have these different parts to our T cell. The CD4 will actually bind to the MHC2. And there's two things then that are occurring here. Number one, we're recognizing a foreign agent or some sort of antigen. And at the same time, we're recognizing that it's being presented by a cell that is our own. And this is the key part of this. We want to know that it is being presented by one of our own cells, and then also recognize what part of it is foreign. And at this point, the APC then, that antigen presenting cell, will activate the T helper cell. It will also secrete interleukin 1, and the T helper cell in turn produces interleukin 2. And this is a growth factor which will help grow more T helper cells and cytotoxic T cells. And this then will go on, these T helper cells can then go on to help activate the B cells. And you can see that over here once it's been stimulated. So with this antigen processing and presentation, the activated T cell is going to be central to all of the T cell immunity and most of B cell immunity. It's going to be that catalyst to get those two immunities moving. Most antigens must be presented first to T cells, even though they will eventually activate both the T cell and B cell systems. So this is the end of the third lecture. The next two lectures will go into the T cell response and the B cell response.